So I work with animal models of Alzheimer's disease that recapitulates different aspects of the disease. And the question is where abnormal electrical activity is coming from in animal models. So we know that patients with Alzheimer's disease, they experience seizures, they experience abnormal electrical activity in between seizures, but we really don't know where this activity is coming from. And the reason is that we cannot do invasive recordings in humans. There are some studies with minimal invasive approach, but we really don't have the resolution to where this activity is coming from. So that's why we turned into animal models. We implant the mice with electrodes, and we asked where this abnormal activity in the form of interictal spikes is coming from. So what we found was that a normally silent area is the hotspot of this abnormal electrical activity in 3 different mouse lines that recapitulate aspects of Alzheimer's disease. We also asked why epileptiform activity often occurs during sleep. So we recorded the mice when they are sleeping, where they were awake, and what we found is that similar to humans, epileptiform activity is robust during sleep. And we found that a specific sleep state was the hotspot of this activity. So then we turned to a specific brain area called the medial septum, with the reason being that this area becomes overactive in Alzheimer's disease. So in the mouse, we inhibited that area, and what we found was that epileptiform activity was significantly reduced. And that was very exciting to us because such interventions are not possible in humans, but they may inform treatments for the future. So our research used a pharmacological approach, but we would like to be even more specific, and we're planning to actually do a closed loop protocol to detect the epileptiform activity in real time and disrupt it in real time, so that we have a very specific way to address whether epileptiform activity is coming from this area and how we can selectively intervene. We know that Alzheimer's disease is very heterogeneous, and it is always important to recognize that there are limitations in the animal models, there are limitations in human research, but our perspective is that we use different models to address our questions, to address whether the results are generalizable in different models. And there's so many models that we can record the EEG from, but we really would like to compare and contrast what we find in animal models and what we find in humans, what is common, what is not common, and how we can all work together with clinicians, basic scientists, and allied health professionals to develop targeted treatments that are going to help people with Alzheimer's disease and epilepsy.